Hello students, in today's module we will learn about the ABC superfamily of transporters. Among the ATP powered pumps that transport metabolites across membranes, the ABC superfamily of transporters is a large family of multiple members that are more diverse in function than those of the other classes. The ABC or ATP binding cassette superfamily includes several hundreds of different transport proteins found in diverse organisms ranging from bacteria to humans. The first of these proteins was characterized in bacteria as transport ATPases in the bacterial plasma membrane and are called ABC transporter superfamily because each member contains two highly conserved ATP binding cassettes. In E. coli, about 80 genes which constitute 5% of the bacterium's genes encode ABC transporters. The human genome includes more than 150 ABC transporter genes. Discovery of the first eukaryotic ABC protein to be recognized came from studies on tumor cells and cultured cells that exhibited resistance to several drugs with unrelated chemical structures. Such cells were eventually shown to express elevated levels of a multi-drug resistance MDR transport protein originally called MDR1 and now known as ABCB1. This protein uses the energy derived from ATP hydrolysis to export large variety of drugs from the cytosol to the extracellular medium. The MDR1 gene is frequently amplified in multi-drug resistant cells, resulting in large overproduction of the MDR1 protein. The substrates for mammalian MBCB1 are primarily planar lipid-soluble molecules with one or more positive charge. They all compete with one another for transport, which suggests that they bind to the same or overlapping sites on the protein. Many drugs transported by the ABCB1 diffuse from the extracellular medium across a plasma membrane unaided by transport proteins into the cytosol where they block many cellular functions. Two such drugs are colchicines and vinblastine, which block the assembly of microtubules. ATP-powered transport of such drugs reduces their concentration in the cytosol. As a result, a much higher extracellular concentration of the drug is required to kill those tumor or cancer cells that express ABCB1 than those that do not. A related and equally sinister phenomenon occurs in protist parasite Plasmodium falciparum, which causes malaria. Plasmodium parasites, which have developed resistance to the anti-malarial drug chloroquine, have been found to have an amplified gene encoding an ABC transporter that pumps out chloroquine out of the organism's system. The control of malaria is hampered due to the development of these resistance to the drug by mutant parasites. In yeasts, an ABC transporter is responsible for exporting a mating pheromone across the yeast plasma membrane. In most vertebrate cells, an ABC transporter in the endoplasmic reticulum membrane actively transports a wide variety of peptides during antigen processing and presentation. The peptide fragments are displayed on the cell surface for scrutiny by cytotoxic T lymphocytes, which will kill the cells if the peptide fragments appear to be foreign, like viral proteins if the cells have been infected by a virus. About 50 different mammalian ABC transporter proteins are now recognized under 7 families. Several are expressed in abundance in the liver, kidney, and intestines. These are sites where toxins and waste products are removed from the body. The molecules that are transported by these transporters are sugars, amino acids, bile acids, phospholipids, peptides, proteins, toxins, and foreign substances. The normal function of ABCB1 is most likely to transport various natural and metabolic toxins into the bile or intestinal lumen for excretion or into the urine from the kidneys. During the course of its evolution, ABCB1 appears to have acquired the ability to transport other toxins or drugs which are similar to the endogenous toxins. As a result, tumors such as hepatomas, which are liver cancers, are difficult to treat because they exhibit increased expression of ABCB1 or a related ABC protein, and thus makes them resistant to almost all chemotherapeutic agents. Now let's study the structure and transport mechanism of ABC transport proteins. ABC transporters have a characteristic architecture which consists minimally of 4 domains. 2 transmembrane domains or TMDs embedded in the membrane bilayer, and 2 cytosolic domains designated as the nucleotide binding domains or NBDs located in the cytoplasm. At the sequence level, the superfamily of ABC transporters is identified by a characteristic set of highly conserved motifs present in the NBDs. In contrast, the sequences and architectures of TMDs are quite variable, reflecting the chemical diversity of the translocated substrates. Beyond these 4 domains, additional elements may be found fused to the TMDs and or the NBDs of the ABC transporters that likely serve regulatory functions. For prokaryotic ABC transporters that function as importers, substrate translocation is also dependent on another protein component, a high affinity binding protein that specifically associates with the ligand in the periplasm for delivery to the appropriate ABC transporter. Each of the TMDs have several membranes spanning alpha helices and form the pathway through which the transported substance crosses the membrane. They also determine the substrate specificity of each ABC protein. The TMDs of a typical ABC transporter are believed to contain a total of 12 transmembrane alpha helices, that is 6 per domain, but in reality the membrane spanning subunits are structurally heterogeneous with 3 distinct sets of folds currently recognized. These folds have been designated as type 1 ABC importer, type 2 ABC importer, and ABC exporter. However, the sequences of NBD domains are approximately 30 to 40% homologous in all members of this superfamily, and this indicates a common evolutionary origin. The 3-dimensional structures of ABCB1, together with those of the homologous bacterial ABC transporters, showed its mechanism of transport of a wide array of hydrophilic and hydrophobic substrates. The mechanism of active transport by these proteins is as follows. The catalytic cycle begins with a transporter free of both ATP and substrate. The transporter can interconvert between the closed and open forms. Then substrate enters the central cavity of the open form of the transporter from inside the cell. Substrate binding induces conformational changes in the NBDs or the ATP binding cassettes that increase their affinity for ATP. Then ATP binds to the ATP binding cassettes or NBDs, changing their conformation so that the two domains interact strongly with one another. Then the strong interaction between the ATP binding cassettes induces a change in the relation between the two membrane spanning T domains or TMDs, and this leads to release of the substrate to the outside of the cell. The hydrolysis of ATP and the release of ADP and inorganic phosphate reset the transporter for another cycle of transport. So as the transporter cycles through the stages of ATP binding, hydrolysis, and product dissociation, the interface between the two NBDs switches from a closed interface with the bound ATP sandwiched between the conserved sequence motifs of different domains to a more open interface characteristic of the non-ATP states. These nucleotide-dependent conformational transitions ultimately drive the conformational changes in the transmembrane domains or TMDs, resulting in substrate translocation. Eukaryotic ABC transporters generally act to export molecules from inside the cell. On the other hand, the prokaryotic ABC transporters generally import molecules from outside the cell. A specific binding protein generally acts in concert with the bacterial ABC transporters, delivering the substrate to the transporter and stimulating ATP hydrolysis inside the cell. Thus, ABC transporters use a different mechanism from the P-type ATPases to couple the ATP hydrolysis reaction to conformational changes. Nevertheless, the result is the same. The transporters are converted from one conformation capable of binding from one side of the membrane to another that releases the substrate to the other side. Now let's study the flippase activity of ABCB1 protein. ABCB1 can move or flip a hydrophobic or amphipathic substrate molecule from the inner leaflet of the membrane to the outer leaflet. This is an otherwise energetically unfavorable reaction powered by the coupled ATPase activity of the protein. Support of this flippase model of transport by ABCB4, originally called MDR2, which is homologous to ABCB1. ABCB4 is present in the region of liver cell plasma membrane that faces the bile canalicule. It moves phosphatidylcholine from the cytosolic to the exoplasmic leaflet of the plasma membrane for the subsequent release into the bile in combination with cholesterol and bile acids. This cholesterol and bile acids are also transported by other ABC transporters. Several other ABC superfamily members participate in the cellular export of various lipids by mechanisms which are presumed to be similar to that of ABCB1. ABCB4 was first suspected of having phospholipid flippase activity because mice with homozygous loss-of-function mutations in the ABCB4 gene exhibited defects in the secretion of phosphatidylcholine into bile. The ABC cystic fibrosis transmembrane regulator, or CFTR. Several human genetic diseases are associated with defective ABC proteins. The best studied and most widespread is cystic fibrosis, caused by mutation in the cystic fibrosis transmembrane regulator, CFTR, also called ABCB7. Like other ABC proteins, CFTR also has two transmembrane domains and two cytosolic ATP binding domains. CFTR contains an additional regulatory domain on the cytosolic phase R. R links the two homologous halves of the protein. CFTR is not an ion pump but a chloride channel. It is expressed in the apical plasma membranes of epithelial cells in the lungs, sweat glands, pancreas, and other tissues. For instance, CFTR protein is important for reuptake of chloride ions lost by sweating into the cells of the sweat glands. The chloride channel of CFTR is normally shut. Channel opening is triggered by protein phosphorylation and by binding of ATP to the ATP binding domains. About 2/3 of all cystic fibrosis disease cases can be attributed to a single deletion mutation in the CFTR gene, leading to the deletion of phenylalanine 508 at the ATP binding A1 domain. At body temperature, the mutant protein fails to fold properly and to move to the cell surface where it is normally located. So let us conclude with the following points. ATP transporters use the binding and hydrolysis of ATP to power the translocation of a diverse assortment of substrates, ranging from ions to macromolecules across membranes. ABC transporters function as either importers, bringing nutrients and other molecules into cells, or as exporters that pump toxins, drugs, and lipids across membranes. Members of the ABC transporter family are present in organisms from all kingdoms of life. While exporters are found in both eukaryotes and prokaryotes, importers appear to be present exclusively in prokaryotic organisms. ABC transporters constitute the largest protein family in E. coli, including about 80 distinct systems representing 5% of the genome, while about 50 ABC transporters are present in humans. Members of the 7 families of human ABC transporters participate in cholesterol and lipid transport, multi-drug resistance, antigen presentation, mitochondrial ion homeostasis, and the ATP-dependent regulation of ion channels. Mutations of these proteins have been associated with a range of disorders, including cystic fibrosis, hypercholesterolemia, and diabetes.